Yes.
Okay, so good afternoon, everyone. It's a pleasure to be here. I will talk on Nematode Holobiont and role of microbes in nematode suppression and protection and some of the work we have done over the years. So this slide is just to show you the importance of nematodes in the soil food web. So there are 4 times 10 to the power 20 nematodes in subsurface soils that play various functions in the soil food web. So we have these microbivore nematodes that feed on bacteria and fungi and release the nutrients for the plant uptake. Then we have plant parasitic nematodes that feed on the plant roots, but also serve as a food for higher trophic groups. We have predatory nematodes that feed on other nematode groups and also serve as a food for higher trophic groups, and some omnivores that have various food. So during my research, I have been focused the most on plant parasitic nematodes, and in particular these root knot nematodes on the left side. They have a specific life cycle. So this J2 stage, or the second stage juvenile, they hatch from these eggs in soil, they infect the roots, and then during feeding they become sedentary and eventually develop into these fallen females that produce eggs in egg masses. So this is the figure from our review from a couple of years ago, where we wanted to show how microbes can suppress nematodes. And so, for instance, while juveniles are in soil, this is the only stage that can be present in the soil. So microbes attach to the nematode surface. And important to mention here is that most plant parasitic nematodes cannot ingest the microbes, but they, because they have very narrow feeding apparatus, so microbes are attached to their surface. And so some microbes can parasitize nematodes right here, but also nematodes can bring the microbes inside the roots, and these microbes can induce systemic resistance in plants against further entrance of the nematodes. Also, microbes can parasitize eggs or females, and also they can produce volatiles or some secondary metabolites. And I have worked a lot also with nematode suppressive soils, and in particular, I was interested to see which microbes associate with the surface of these second stage juveniles in nematode suppressive and nematode conducive soils. And so what we found was that in nematode suppressive soils, such as this soil G from Geisenheim and BS1 from Dansdorf, so both soils, all soils from Germany, bacterial profile was more diverse on these juveniles compared to some non-suppressive soils, such as these KWS or JKI. And also some genera, such as this Devosia or Rhizobium, were shared among nematodes in nematode conducive soils, and we propose that they may have some facilitating role for the nematodes. So we know that there are microbes that suppress nematodes, but what about the microbes that may protect them? So for plant parasitic nematodes, this has not been studied. And we proposed this in our opinion article a couple of years ago, that there are microbes that can actually facilitate nematode parasitism and their survival in soil. And this is, again, the life cycle of these root knot nematodes. There is second stage juvenile in soil. Then in roots, there are developing females and egg masses with eggs. And so we propose that during different life stages, nematodes acquire specific microbes on their surface that may have some function. So this includes also the galls inside the roots that nematodes cause by their feeding. So when these juveniles hatch in the soil, there may be microbes that protect them against some antagonists or also facilitate their infection into new roots. And so, for instance, here we explored microbial communities that associate with nematode galls, healthy roots as well, egg masses and females of root knot nematodes on different plant hosts. And we found that plant species had a higher effect on fungal communities compared to the origin, so to these samples. And this CAP1, it explained for, I think, 44% variation and CAP2, 24% variation. And we also noticed that there was a sort of a clustering of fungal communities for the gall samples, which suggests that there may be some similarity in these fungal communities. We also explored how bacteria associate with second stage juveniles in soil and whether there are some protective taxa. So we surface sterilized these juveniles and exposed them to different treatments. That was either PBS as a sterile control. Then we also used bacterial strain Pseudomonas protegens cow. We also extracted microbiomes from 2 soils, these G. Geisenheim and F. is Flakeberg. And also, additionally, we amended these soil suspensions with bacterial strain Cal. And after 3 days, we counted number of dead or non-motile and live or active nematodes. And we found that actually when we add this bacterial strain to the soil suspension, the nematodes were more active. We also collected these active and non-motile nematodes to study which bacteria associate with their surface. And for instance, here on the left side, you can see bacterial ASVs associated with active and non-motile juveniles in this soil G. And on the right side is the case when bacterial strain Pseudomonas protegens Cow was added to the suspension. And so we found that, for instance, on active nematodes, there was an enrichment of this ASV Algoriphagus terigena and Delovibrio. And for instance, in C. elegans, these bacteria were found to confer resistance to nematodes against some antagonists, while on non-motile nematodes, we saw an enrichment of this Pseudomonas ASV, and Pseudomonas is known as a nematode antagonist. We also found that when we add this cow strain to the soil suspension, there are changes in the composition of nematode-attached bacteria. So, for instance, Pseudomonas and Delovibrio, they disappear from the nematode surface. And this is just to show you how the complexities of microbial interactions in soil affects not only the activity of nematodes, but also the composition of microbes on the nematode surface, and that there may be some taxa that are nematode protective. And this slide is just to show you as well, this is scanning electron micrograph of surface sterilized nematodes, so there are not microbes on their surface. And this is when this Microbacterium strain K6 was attached to the nematodes. And finally, I would like to thank my collaborators over the years, and also Novo Nordisk Foundation for funding my current projects, and also for giving me opportunity to present my work. Thank you.
Thank you very much. It's very nice to hear about nematodes for a change. We have time for a question or two. Anyone want to have one? Yes, yours.
Yeah, very nice talk. Also, especially with the bacteria that protect the nematodes. I was curious. Do you know what's in it for the bacteria to the nematode?
So we haven't really explored the mechanism, only the composition of microbes, but for instance, nematodes can carry these microbes inside the roots and maybe provide environment for feeding, or also they can carry them from one distance to another, or maybe have no role at all. We are not sure.
We have time for one more if it's very quick. No? I guess a quick one from me. I mean, ultimately, do you think this could be used in sort of control strategies?
Yes, so we propose that antagonists are not so effective in soil because nematodes may carry these protective microbes. And if we understand how they interact with each other, maybe that could improve some or increase success of the antagonists.
So how far off do you think that is? That's a near goal, or that would be quite a way yet?
I'm not sure. So probably quite a way to understand this and also to apply this knowledge in the field. Probably may take some time, but yeah, we are optimistic.
Great. Well, thank you again.
Thank you.